Hello and welcome to BioCode. In this video, we will see how we can utilize sequence IO module to extract various amount of data from a particular GenBank file. We will parse it into sequence IO module, and then we will extract all kinds of information that we have been learning throughout this series. So this is a particular GenBank file that has multiple GenBank records within this particular file, and they are separated through these backslashes, and they are actually uh, quite a lot of sequences available within this particular GenBank file. So we can do all kinds of extraction on this. We can extract features, we can extract annotation, IDs, names, descriptions, annotations, features, whatever information that we can extract from this particular GenBank file. So let's go back to our original Python code, and in this Python code, what we are going to do is we are going to first of all demonstrate that how we can extract all kinds of ID, sequence, or that sort of information. So let's utilize to list. First of all, I'm going to first of all create a list of IDs, and let's make it separate. Let's also create a list of sequence, and then we can store our all of the sequences, all of the IDs in separate lists. So first of all, we are parsing this particular file, that is GenBank file, and we are telling that it is a GenBank record or GenBank format, and then we are parsing over each of the record. Let's go back, and now we need to do is list of IDs is dot append, and then we need to pass in the record.id parameter. On the next, we need to pass in the record.seq, and just make sure that it is 6 on this end. So it is pretty much that it, and once the entire loop is executed, we will print out the list and of IDs and list of sequences. So let's run our code and see if there is an issue with it or it is executed like this. So as you can see, first of all, we see that let's print them out separately, so there's no confusion. And there we go. Okay. So first of all, what we see is the list of IDs, all of the list of IDs, and they are all different records, and they are in a single list. And then we have the single list record in a single list. So let's make it like this, and representation, and now we can execute it. There we go. So this was pretty much easy to just extract the sequences or extract the IDs. What more information can we extract from this particular set of code? I'm going to keep it like this. You can find this code in the transcription, and I'm going to run another loop over the same file. So just to keep it simple for you guys, so that it does not become more of a complex thing. So now we can also extract the features, and as you know, we can extract features of our GenBank records. And first of all, I need to do this, and let's run our code, and then you will see all of the features of entire of the file will be printed out like this. Now they will be printed out as separate list. For example, each of the sequence has its own features, right? So each of the feature of a particular sequence will have its own list, and they will be printed out separately. And we can also run a data mining or data extraction technique on this sort of information. Let's extract the annotations, and let's see what prints out. Then we will extract the pattern wise information from that sort of result. Okay, so these are the annotations of a particular sequence. Let's try to because this file contains multiple organisms, data of multiple organisms or multiple species. So what I'm going to do, I'm going to extract all of the information that has this organism, right? And that is extracted of this organism. So let's go back to our original code. I'm going to keep it as a record over here. Then I'm going to pass as organism. So we need the organism information. We don't need any other information, right? We just need the organism name. And what will happen now is that when I execute this code, only the information that is related to the organism will be printed out. Let's go back to our original result. As you can see, this was the initial annotation, and in that initial annotation you can find a particular key in this dictionary. Basically this is a dictionary of lists, right? And we can find source or organism key that says, okay, this is the particular organism of this particular sequence. So if we pass in the organism key to that annotation dictionary, it will print out just the organism name or the source name. So now we can run our if else statement and we can extract that particular information. If record organism is equal to and print record dot annotations, or so we can just print out the IDs now. If the annotation has a organism name of this organism name, then we can just print out their IDs. So let's print out their IDs now, and so that means only a single organism in this entire file has this name, and only that ID is printed out. Let's make sure that it is indeed true. For the sake of demonstration, I'm going to utilize some sort of different name that has multiple, so that we can extract info. Okay, this seems to have multiple results, and let's utilize them, and there we go. Now let's rerun our code. We will now see multiple IDs. There we go. We now see multiple IDs. So this was pretty much easy. This is how you can utilize the code to extract information. It depends what are the questions that you are asking to your data and what you would like to extract from it. So you can also run now you can print out the sequences as well. You can print out the descriptions as well. Let's print out the descriptions as well, and that will be more of useful for us. You can directly write them out into a separate file, as you